Data exists everywhere. Is data limited to numbers and words? No. In the field of microscopy, images are the data. Today, we are going to show you a novel image analysis workflow to segment bacterial cells from 3D dense biofilm data. Our project is visualizing cellular behaviors in bacterial biofilms using Lattice light sheet microscopy. Bacterial biofilms is a collective of bacterial cells aggregated on solid surfaces. It plays critical roles in human health and the natural environment. Understanding individual cell behaviors within biofilm is a stepping stone to either harness or prevent biofilm growth output for human benefits. But it's really hard to study individual cells in dense biofilm due to the lack of suitable imaging and image analysis techniques. To overcome these barriers, we proposed our workflow that incorporates cutting-edge imaging technique with novel data processing algorithms. The first challenge in the field of biofilm study is about imaging methods. Conventional imaging methods, including confocal and wide field microscope, have high phototoxicity due to their high illumination intensity on the sample. The high phototoxicity can impact the growth of bacterial biofilms. Therefore, these conventional imaging methods are not suitable to image live bacterial biofilms for long term. Comparing to conventional imaging methods, Lattice light sheet microscope have higher resolution and lower phototoxicity. The resolution of Lattice light sheet microscope is in the range of 200 to 300 nanometer, which is smaller than the size of bacterial cells. Therefore, Lattice light sheet microscope can image bacterial biofilm with subcellular resolution. In addition, the illumination intensity of Lattice light sheet microscope on the sample is comparable to sunshine. The low phototoxicity make it suitable to image live bacterial biofilms for long term. In our lab, we construct a Lattice light sheet microscope and apply it to study bacterial biofilms. The data we collected with Lattice light sheet microscope are 3D images. Here are some examples. Those are 2D slices of the 3D datasets. For a long-term experiment, several hundreds of gigabytes datasets are usually collected. To process these large datasets, an automatic and robust imaging analysis method is required. And the second challenge in the field of biofilm study is about image processing, in specific, bacterial cell segmentation.
With the data and the challenges in the data, there are frequent cases of over-segmentation and missing cells in the current toolkits in the biofilm community. To achieve better segmentation accuracy, we propose a workflow as shown, which mainly consists of two analysis parts. One is using convolutional neural networks, UNet, to get in silico transient preliminary outputs. Then, we post-process the outputs to further analyze unsolved cases to get the final output. The data for training the CNN are simulated 3D biofilms that the experimental acquired point spread function, PSF, is convolved with the simulated cell arrangement. Different biofilm cell densities and background noises are also generated to test the performance for different experimental settings. The post-processing module is realized by three major steps. The first step will filter out the noises produced by the in silico transient and select the under-segmented clusters for further analysis. Then, we generate the point cloud data that lies on the media axis of these clusters using in-scale spheres with constant radius. These point cloud data will then be further splitted into different groups recursively. where single cells are identified. Finally, the biofilm can be reconstructed with the output from LCuts by fitting shape models or enclosing the enclosed spheres found earlier in a convex hull to refine the cell surfaces to better align with the thin segmented volumes. We evaluated the performance of proposed workflow with 1,000 simulated datasets with 10 signal-to-background ratio conditions and 5 density conditions. The evaluation matrix we're using here is cell counting accuracy. If the experimental result has an overlap area with ground truth that is larger than a thresholding value, then we will count the current result as a successful segmentation count, which is true positive. We notice that high density of the cells in the biofilm is a key challenge to the in silico trained neural networks. The cells are still connected in the preliminary result. Quantitatively, only 37% and 59% cell counting accuracy are achieved for low SBR and high SBR images, respectively. However, compared with the other methods in the biofilm community, the results before post-processing can already consistently achieve better performance. You can see from the 3 figures on the upper right. These are selected comparisons with different SBR and density conditions, with Jacquard index threshold vary from 0.1 to 0.9. Lower 3 figures show the comparison of after post-processing using LCuts with before post-processing under same SBR and density conditions. After post-processing, improvements of up to 36% are observed over the 1,000 data. We also evaluate the proposed method on real experimental data. However, due to the difficulty of getting ground truth in 3D volume, we evaluated the result in 2D slices. And again, results after post-processing using LCuts achieve the best performance where connected cells are separated. In conclusion, we presented a dataset produced by a cutting-edge imaging technique that is able to perform long-term live cell imaging with single-cell resolution in 3D. We also introduced our proposed image analysis workflow for this dataset, which can consistently outperform the other methods. It can also help with cell tracking in dense 3D biofilms for the future work. You can find more details about our work on bioRxiv. Finally, we would like to express our gratitude to our advisors and labs, and School of Data Science for supporting our collaboration. Thanks for listening. Have a good day.